It is my distinct pleasure to introduce Dr. Carissa Chu. She is one of our own. She did her medical school here at UCSF and residency here too. And during her research year, she was a recipient of the TL32 training grant and spent her year in the lab with Dr. Felix Feng and Dr. Jonathan Chu. She completed her fellowship at Memorial Sloan Kettering and is now back here with us. Just started in September as an assistant professor here in our department. She's a true surgeon scientist. She has her wet lab over here in Diller, and I just got to meet her team, including her postdoc and everything, and they are up and running. And yeah, we were really, really excited to have Dr. Chu back with us, and I will let you get started with your presentation.
Thank you so much, Dr. Porton. Oh man, save it for the end. Just kidding. So good morning, everyone. I'm just so excited to be here and have the opportunity to talk about, what I've been working on these past few years, what I hope to be working on for the next 30 years or so. So I know we have kind of a wide audience here, so I hope to keep this, we'll talk about the science, we'll talk about, stuff that I have planned for the lab, but try to keep a kind of high level overview of what we're trying to accomplish here. Can people here on the Zoom hear us okay? Okay, so our presentations aren't synced because of some technical issues, so just please bear with us. So the goals of this talk, first I want to go over the describe advances in the treatment of metastatic urothelial cancer. So that's sort of the what. I want to identify underlying mechanisms of resistance to improve our patient selection and identify other therapeutic vulnerabilities. So that's the why. And lastly, I want to highlight the current progress, experimental approaches, and the unique team we have here at UCSF to accomplish some of those things. And so I want to start by talking about Enfortumab vedotin. This is a drug hopefully the residents are familiar with. It's now testable on the in-service, which means it's here to stay. But EV is a novel therapy in the treatment of urothelial carcinoma. It's an antibody drug conjugate. And what that means is, so if you can see, I guess you can't see my pointer there. So it's an antibody that targets Nectin-4. Nectin-4 is thought to be ubiquitously expressed on urothelial cancer cells. And when it binds, it's internalized by the urothelial cell, cancer cell, processed by lysosomes, releasing this payload, or MMAE. And MMAE has been around for a very long time. It's a microtubule destabilizing agent. And so it's the antibody with the linker to the payload. The whole drug is internalized process, and that leads to cell death and apoptosis. So next slide, please. You see it? Oh, it's not showing here. Okay. All right, so EV-201 is the pivotal trial of Enfortumab vedotin is really what brought EV into the field, and this was published in 2019. And this was a single-arm phase 2 study looking at EV in the post-chemotherapy, post-immunotherapy setting. So these are heavily pretreated patients that have progressed on all standard of care therapies. And what you can see is that, over on the left, so this is a swimmer's plot of patients who responded, okay? And so the blue line is how long their response is. The red dot here on the left shows, at what point there is some sort of radiographic response. And you can see that, in these patients who otherwise have very few other alternatives, this is a marked response, and this was seen to be really a groundbreaking therapy for these patients. Over here on the right, you have what we call a waterfall plot, and it shows sort of the best radiographic response evaluable. So they take the diameter on surveillance imaging for these patients on treatment, and they measure add all that up, and they say over time how much has changed. And so when the waterfall plot goes down, that's a good sign because that means the tumors are shrinking. And so this study showed a 44% overall response rate in, again, these heavily pretreated patients, 12% complete response rates. And so this expedited approval of EV in the third-line metastatic setting. And this is their table one, essentially showing that 100% of these patients had received some form of immune checkpoint inhibition. 100% of them had also received some prior platinum-based therapy. So again, these are patients that are fairly, have progressed quite a bit on their disease. And this is a supplementary figure from an earlier study. It was the dose escalation trial, showing how prevalent we think Nectin-4 expression is in these patients. And this is by immunohistochemistry. And an H score is something our pathologists read out, and it's a composite score of how strong that area is staining positive for that target, so 1, 2, or 3, and times the percentage of how positive that is. So if it's really strong staining on 100% of the cancer cells seen, then that gives you a 300 H score. So it goes from 0 to 300. And this table from their early data shows that Nectin-4 expression looks like it's somewhat somewhere in the ballpark of 97%. So very, very strongly expressed in pre-treatment tissue. And so fast forward a few years, 3-4 years later, and this is a study that was just presented at the European Society of Medical Oncology, or ESMO, in October of this year. And so EV has now moved to the frontline setting. And this is a clinical trial showing EV plus immune checkpoint inhibition, so pembrolizumab, compared to standard of care gemcitabine plus platinum-based therapy. And that's the standard of care. You guys all know that that's the frontline treatment for patients with metastatic urothelial carcinoma. It's gemcitabine and cisplatin or carboplatin, so some form of platinum-based therapy. And this is now shown to be superior to that. So it's really only a matter of time before EV plus pembro will replace traditional chemotherapy in the first-line setting for metastatic urothelial carcinoma. And so the blue line up top is the EV plus P cohort. And then the gray line is, they make it look not as good, but it's chemotherapy, which is standard of care right now for our patients. Quite a few patients, close to, looks like, close to 900 patients there with a hazard ratio of 0.45. So again, very, very impressive response rate with a very long tail of response that you can see there. And so the overall response rate here is 67.7% versus 44%. So again, significant superiority. And the overall survival is now extended about double. So it used to be 16 months and now 31.5. So when I look at this graph, there are a couple of questions that come to mind, okay? And so, is this a synergistic effect between EV plus pembrolizumab, or could these two agents be kind of acting on different subpopulations that a patient may have, or both. And you can imagine kind of some time in the future where if treatment fails patients on traditional chemotherapy or treatment fails for patients with EV plus pembro, they probably will cross over to the other form of treatment. So does it stand, is it possible to think about, are there fundamentally different things about these patient tumors that render them more susceptible to one treatment or the other? Because it's not that nobody is responding to chemotherapy, right? And there's likely a continued role for chemotherapy in these patients. So, which cell populations are responding? Is there a way for us to predict who will respond to what? And how much synergy is there between the immune system, the immune microenvironment, and some of these new novel therapies? And the other thing to consider is that, as is the case in so many other disease states, when something works in the third line, it gets moved to the first line. When it works in the first line, it gets moved to the adjuvant line. When it works in adjuvant, it then gets moved to neoadjuvant. So these treatments are coming to us, and our treatments will be on it. Our patients will be on it when we take them to surgery. And so I think so we started the, pretty simple question here. This is work, this was part of my research here as a fifth year. So if you were here in the room for that, apologize for showing these slides again. But we just really wanted to look at what is the heterogeneity of Nectin-4 expression in muscle invasive bladder cancer, okay? And so these are 4 publicly available cohorts. And so they have transcriptomic data that's already available that we can look at, Okay, and so what we did was we applied this consensus molecular classifier. And what a consensus molecular classifier is you look at all the genes that are expressed in that tumor, and it seems to sort of cluster into these different subtypes. Okay, and there's actually 6 that have been described. And Dr. Porton's fellowship at ND Anderson was really integral in kind of coming up, developing the basis for these signatures. And so, kind of high level here, I've kind of just shown in the center there, there's the basal subtype, there's the luminal subtype, which there are actually a few of them, but I kind of lumped them together as one for the purpose of this talk. The neuroendocrine subtype and a stromal subtype. And what you can see circled in orange is that Nectin-4 expression seems to be highest among the luminal subtypes of bladder cancer. And again, this is based off of published cohorts, that were studied for other questions. We just looked at the transcriptomic data. And in fact, when you take this into the lab and you look at different cell lines that also are categorized now as basal or luminal, because we do have the gene expression for, all of these cell lines have been sequenced and categorized. Generally, you can bucket them into, are they basal or are they luminal? And what you can see on this Western blot here is that when you stain for Nectin-4, the luminal subtypes of bladder cancer appear to be enriched in Nectin-4. And in fact, when we just take those different cell lines and we expose them to EV, what you can see is that this is a dose response curve. So for increasing doses of Enfortumab vedotin, the red and the orange, those cell lines are dying off more quickly at lower doses, so they're more sensitive to EV. The blue and the black lines are basal lines, and you can see it takes higher concentrations for them to die off. And so, without doing any sort of manipulation of these cell lines, you can see that there's differences in sensitivity to this drug. And that's based off sort of their basal and luminal identity. And then when we play around with Nectin-4 levels, we can actually mediate the response to EV. So HT1376 is a luminal cell line, okay? And so you can see that in red here, it's very sensitive to EV at very, very low concentrations. If we then take that same cell line and remove all the Nectin-4 expression through a CRISPR platform, we can actually induce some resistance there, which, eventually they do die off, and you can give toxic enough levels of drug. Probably that may be an MMAE mediated effect where you're just giving so much chemotherapy that they're dying off. And this is sort of a cell, cell viability assay here, showing that the control cells are dying off at these very low concentrations, but our knockdown cells are kind of persisting through it. And then so you can do the opposite. Then we take these UMUC3 cells, which are basal cell lines, then they have no basal Nectin-4 expression. You induce overexpression of Nectin-4, and that's that orange line here, where now they're very, very sensitive to EV. These very low concentrations of EV are knocking out, are killing these cells early. And, they're probably really overexpressing Nectin-4 because very, very low concentrations are very toxic. And you can see that in the cell viability assays down below. So this has been published as of a couple years ago. And the other thing we showed was that what we did was we took the HT1376s, again, the luminal cell line, and we exposed them repeatedly to EV. And over time, what you can see is that dose response curve shifts up, right? So over time, these cell lines are becoming more resistant to EV. And when we look at the Nectin-4 expression by Western blot, you can see that these cells are actually turning down the expression of Nectin-4. So it's kind of a nice, simple story showing that when Nectin-4 is both necessary and sufficient for EV mediated cell death, and in a functional way, when the cells are exposed to EV, one of the ways that they develop acquired resistance is by regulating the antigen or the epitope of interest. And so part of the work that I did in fellowship was to see, are these observations true in patients? Can we actually look at whether or not, Nectin-4 or other possible predictors of response are observable in patients? And so Memorial Sloan Kettering has one of the largest cohorts of patients treated with EV due to, Dr. Rosenberg and others who have led this, international clinical trials on this work. And so this is a cohort of, it started around 130 patients who had been treated with EV monotherapy, again, in the third line setting. And what I want to show here is that when we look at Nectin-4 expression in their pre-treatment tissue, and that can be a TUR, that can be a radical cystectomy, there's a strong correlation between Nectin-4 expression and response to therapy. Okay, so PD stands for progressive disease, those are the red dots. Any sort of response, partial or complete response, are the blue dots. And the y-axis is the Nectin-4 H-score, as determined by our pathologists. And, there's a statistically significant difference there in the baseline Nectin-4 expression and the patients who do and don't respond to EV. Pictures are kind of worth 1,000 words there. So, on the right is the immunohistochemistry staining for Nectin-4. So anything that's Nectin-4 is brown. You could see that patients who've got higher Nectin-4 staining and expression, those are examples of how they're responding. And UCNOS, your standard, urothelial carcinoma, as well as a micropapillary tumor. We think of some of these variants as having more aggressive histology. That this one had preserved Nectin-4 expression, and this patient responded very well. That's in contrast to small cell neuroendocrine, which is at the bottom there, which is a pretty aggressive variant as well. These tumors really don't stain well for Nectin-4, and it's been our observation that these small cell neuroendocrine tumors probably don't respond to EV monotherapy very well, and that was given an H-score of 0. So this is also some published data of a very, very nice study that was published earlier this year by this group doing something very similar, looking at Nectin-4 H-scores and associating that with clinical response to EV. And then here they have there was a cohort of 47 patients with treated with EV monotherapy. And, they kind of picked this cutoff of 100, and I'm not quite sure where that 100 came from, but you see a very nice separation in the survival curves there of sort of these high Nectin-4 and low Nectin-4 expressing patients. Okay, so again, it looks like Nectin-4 may be associated, at least in a retrospective way, with EV response. And then this is also kind of going back to, this sort of luminal and basal observation, which is that when we take a TMA, so some of these tumors that are, kind of taken aside for further staining, further correlative studies, there's a range of Nectin-4 expression there from tumor 1, tumor 2, tumor 3. And tumor 3 is a luminal cell line, a patient with a luminal tumor. Again, looking at a number of different patients there on the TMA that have Nectin-4 staining, these luminal subtypes also have very high Nectin-4 expression. The other interesting thing is that Nectin-4 expression is highly variable across different bladder cancer states. And so this is no longer, these aren't treatment patients. This is just what is Nectin-4 expression in non-muscle invasive disease and invasive disease and so on. And so this is a high risk papillary tumor. It's actually a cystectomy specimen. And not only does this tumor stain very strongly for Nectin-4, you can see there's a very large membranous fraction of it. So a lot of it is actually present at the surface, and we think that may be important for targeting of the drug itself. Contrast that to CIS here, and so CIS is sort of like this one has sort of like this pagetoid spread, and I think this is actually a ureter. What you can see is that, there's sort of like this crawling appearance, the CIS, and you can see the parts that are highlighted by Nectin-4. And there seems to be, if you compare it to the normal urothelium next to it, there seems to be something that's happening where Nectin-4 is then trafficked to the cell surface. And so that's something worth noting. And again, really poorly differentiated tumors may or may not express Nectin-4. And so here's a PT3B radical cystectomy specimen with a very low Nectin-4 expression. So again, just highlighting that there is quite a bit of variability, even though sort of the initial preliminary work, the industry-sponsored study showed that there's ubiquitous expression of Nectin-4, there's actually a little bit more variability than we think. This is also highlighting a difference between, a sort of the membranous appearance of it as well as the more cytoplasmic fraction. So going back to that initial paper, now we're talking about heterogeneity and concordance, right? And so in this paper that was also published this year, the authors looked at what is the difference in Nectin-4 expression or localization in the primary tumor compared to a metastatic site, so a lymph node, a lung biopsy, and what the authors found is that Nectin-4 appears to be downregulated at metastatic sites. And you can see that by their violin plot on the left, where it looks like the median expression is going down quite a bit there. And then some representative images to the right of, again, Nectin-4 immunohistochemistry showing that in the metastatic state, there may be some sort of tumor evolution or plasticity which occurs where Nectin-4 is being downregulated. And this has very important implications for, can we really truly use Nectin-4 as a biomarker of predicting whether or not someone will respond to EV. And so this is a commentary that was published earlier this year as well. Again, kind of just highlighting how heterogeneous Nectin-4 expression actually is. This is just a graphical representation of the drug and of the primary tumor, how the drug's acting. But then as it metastasizes to different states, there's a range here, almost a quarter in each group of undetectable versus strong staining. And the change is really actually kind of bidirectional, okay? So maybe 60% of metastatic sites show a decreased Nectin-4 expression, but some sites will actually increase their Nectin-4 expression. And again, that gets to heterogeneity and difficulty with predicting, who will respond to treatment. We kind of talked about different mechanisms of how this might work. And so EV, the first slide I showed was the left side of that direct killing. So how the drug is supposed to work. It's internalized, it releases the payload, it causes cell death. But there's this other hypothesis theory of bystander killing, okay, where a neighboring cell, which is killed by EV, may release some sort of signaling, damps, or even release residual drug itself and cause killing of a neighboring of a neighboring cell, which may or may not express Nectin-4. So there could be some synergy there. And that maybe gets at why the combination therapy is so effective. So the question is really which site matters more, okay? And again, this is going back to the clinical cohort that I got the chance to work with in fellowship. And these are pretty rare specimens. So these are cystectomy specimens that have an accompanying lymph node that's positive. That patient then metastasized, went on to treatment, then had EV. Okay, so there were 10 of those. And if you compare the Nectin-4 expression in the primary and the lymph node, that's column 1 and column 2, you could see there's a little bit of movement there, right? Sometimes it goes up, sometimes it goes down, and this is a very small cohort, so percentages, it's hard to make much of percentages, but it's 50-50. Some go up, some go down. And the associated response to EV there, as well as how long the progression-free survival lasted. And the observation here, and again, these are small numbers, is both the lymph node and the primary tumor Nectin-4 expression were significantly associated with progression-free survival. So even though there's difference from within patients, there may be enough difference across patients that you can still say maybe there's some correlation with response there. So I think it's still an important question we haven't really fully answered there. So conclusions, part 1. So in vitro and retrospective tissue-based studies show that Nectin-4 expression is correlated to treatment response. Okay, but heterogeneity is common, which challenges the use of Nectin-4 expression alone as a biomarker of response, and something that sort of plagues the PD-1 and PD-L1 field as well. So which site matters? Some low expressors may still respond, some high expressors may not. So let's dive a little bit deeper. Let's see if we can answer this a little bit better. And I really want to highlight the work of Kevin Chang here, who is a medical student applying into urology and probably just one of the just gifted, a very mature scientist and did a lot of this work on his own. And, in some ways, in hindsight, I'm lucky I didn't work with this cell line, because the cell line he worked with here, the RT112s, he did something very similar where he exposed them to escalating concentrations of EV. And what he found in this particular cell line is that Nectin-4 expression is not only preserved, it may actually go up in the post-EV setting, okay? And that's the Western blot you see in the upper right corner. The parental or the normal cell lines are kind of low Nectin-4 expressors, but then when you make an EV-resistant cell line, that Nectin-4 expression somehow is going up just a little bit. And so one of the reasons for this, and now this has been, the cell line's been sequenced, is there's an acquired resistance that seems to occur to the payload itself. And that's through upregulation of this drug efflux pump. Probably remember it from medical school, MDR1 or PGP. And the gene that encodes for that is ABCB1. And this is not in bladder cancer, but this is a published study looking at a breast cancer, something very similar in breast cancer. So breast cancer is a potential target for EV as well. What they show is that up here in that top right corner, this is comparing resistant tumors to sensitive tumors, and looking at the differential gene expression between those two. That's how you get that volcano plot. So red means that these genes are really, really much more highly expressed in a resistant state, and you can see this drug efflux pump being way up there in the right corner. It means it's very statistically significant and it's very highly upregulated in a resistant state. So this is a mechanism of resistance in this model as well. Okay, so the protein expression, you can see there's a shift to the right there, again showing that one more slide, showing that the drug efflux pump is actually probably the mechanism of resistance in this model, where the MMAE or the payload is not getting a chance to stay in the cell because it's being actively pumped out. And so what Kevin was able to do, again in his research year with John Chow and others, is to design a CAR T cell. And what is a CAR T cell? So a CAR T cell is a chimeric antigen receptor, okay? It's a genetically engineered T cell, which he made to recognize Nectin-4. And so what you take is sort of that the part that recognizes Nectin-4. There's a heavy chain, there's a light chain, and so he sequenced it. He created a gene and a lentiviral vector, took T cells from healthy donors, transfected them with the gene, which now they express this epitope recognizing domain that will bind to Nectin-4, thereby recruiting the immune system to any Nectin-4 expressing urothelial cells. Next slide, please. And so this is his dose response curve there. So the parental cell line, so the y-axis here is kill index. Okay, so higher bar, more killing. And so the parental cell lines, you can see when they're treated with EV, they're killing, EV is killing it at increasing concentrations. If you then take an EV resistant cell line and you expose it to EV, as you can imagine, you don't see as much killing as you might expect. And so that's why those pink bars are staying relatively flat. But if you then use these engineered CAR T cells, you restore the sensitivity that you had before with EV. Probably, in this particular cell line, which we know has that drug efflux pump. So you're going around an existing described drug mechanism, giving up another way of targeting the same epitope, and restoring the killing that you initially saw. So this is a very important kind of proof of concept here, and a way of mediating drug resistance. And so I just want to highlight John here, who's my mentor when I did my research, when I did my lab year. And he was recently awarded a Beacon YIA for this work. He's the mentor on this project as well as my project. And so this is now part of our repertoire here at UCSF of novel therapeutic drug development. And so next slide here. So, there's sort of this sense that, okay, there are different drug resistance mechanisms described, and what other genes may be altered after response to EV. And so what we did here was, this is such sort of a convenient sample, a swimmer's plot that I put together. So these are patients who were treated with EV, and they all had this pre-treatment, they all have pre-treatment tissue. The green X is where these patients had a post-treatment biopsy tissue. And so this gives us a pre and a post, gives us tissue to then sequence and look at further, and look at, which genes are actually up or down regulated in a post-treatment state. And so these are all the slides these are all the blocks that were pulled in order to do this work. This is just highlighting how crazy this is. So these are selected genes, and these genes are involved in lysosomal processing, they're in lysosomal trafficking, endocytosis, the drug efflux pump is up there, HER2 and TROP2 are other targetable surface proteins. And you can see that there's quite a bit of variability in what happens in a post-treatment state. And so, if the bar goes up, it means it's increased. If it goes down, that means it's decreased. And each bar actually represents a different person. And then I threw at the end there the cell line that we have pre and post sequencing information for. So that gets a little bit messy. We had a number of patients with successful pre and post EV treated tissue, which was, sequenced for their transcriptome. And now we've identified a number of genes that look to be, kind of either very high, upregulated or downregulated in a post EV setting. And so the next step, and this is also showing differences in transcriptomic signature. I'll skip through this here. And then, this is also some work that was done in Dr. Solit, who was my mentor at Memorial Sloan Kettering. So the payload itself matters, okay? And so these are different payloads you can see on the bottom right for different targeted therapies right now. And they act on different parts of, the cell cycle, basically. And DS006 and DS070, these are different organoids. So these are actually patient-derived tumors, which are then brought to the lab and grown. Very, very difficult to do, very time-intensive, resource-intensive. And so if you just take two different cell lines and you expose them to the same amount, same concentrations of the payload, there's a very significant difference in how sensitive they are to these different, chemotherapies. And so in some ways we're maybe chasing our tail a little bit because, sometimes it gets to maybe, is this really just a chemotherapy sensitivity or resistance question? But I think it's very important to keep in mind that when we talk about ADCs, it's not just, the target itself, it's also the payload and how that combination also interacts with the microenvironment. So to do a little bit more interrogation, what we have planned to do next is a targeted CRISPR screen. And so what is a targeted CRISPR screen? It's basically, Luke Gilbert, also affiliated with the department, Dr. Weissman. This is a very powerful tool that's now being exploited in a number of different ways, sort of this functional genomics approach. And so I showed you that graph before about genes that are really heavily increased or decreased post-treatment. So now what we'll do is we'll pull all those genes together and selectively inhibit them, okay? So this Cas9 or dCas9 system is a way of silencing single genes in cell culture. And you can design this in a way that if you have a gene library, you can also do all 20,000 genes if you want. It just takes a long time. You can knock down a single gene in that cell and do it for as many genes as you want. And then you can then expose those cells to treatment, so EV in this particular situation, and see which cell makes it at the end. Okay, so you can see that purple cell on the bottom right. Whatever gene was knocked out made that cell very sensitive to EV, didn't make it to the end of the screen, right? In contrast, the pink cell, whatever gene was knocked down, made it very resistant to EV, and they made it to the end of the screen. And so then what you can do now is then you can look at, who made it, who didn't make it. Talking about cells like they're people, but like what made it, what didn't make it. And then look at, what actually functionally is rendering a cell sensitive or resistant to treatment. And then you have to do a lot of validation after that to say, okay, what pathway is affected here? Why do we think this is important in sort of the mechanism of how EV is working? And so that's one of the first things that we'll look at. This is just some cool stuff. I don't think it'll be a major focus of my portfolio per se, but I think we have a huge opportunity to also look into this at UCSF and collaborate with others. This is work that was done at Memorial Sloan Kettering. And you can imagine now that we have a targetable protein, not unlike PSMA, that there are ways to ligate, radioisotopes to be able to improve our staging imaging and a molecular staging of metastatic bladder cancer, which has been sort of plagued us for a while in persistent clinical understaging of this disease. And so, there's a Dr. Go and Dr. Pilarsetti. Dr. Pilarsetti is a nuclear biochemist, I think. But his lab does this, and we have folks here, Dr. Robert Flavel and others who also do this as well and have developed other molecular targeted imaging agents. So there's a conjugation to a radionuclide. It's injected the same way PSMA is PET imaging is done, and so areas of otherwise occult metastatic disease light up, and this could be a real game changer for patients too. This project has since moved along quite a bit, and been tested in animal models, and it seems to be very, very promising. You could do the same with the fluorophore, so you could mix and match. So same with the Firefly PSMA Firefly study here. Instead of attaching something that will light up on a PET scan, you could attach a fluorophore, which lights up intraoperatively. So this may help with, endoscopic resection. It can help with laparoscopic surgery. And so nice ways to identify sites of active disease. So that's preclinical. It's exciting. It's also another clinical application of some of this work. And so the other thing I want to do is I want to highlight the clinical trial landscape of EV here at UCSF. And, right now, Vadim has a study open looking at EV plus Pembro, which is a combination treatment in bladder sparing, so, putting us out of business. And I think that will be a very, very nice study because it seems to have some immunologic component, and these, that's open here. There's another study opening here on neoadjuvant for high-grade upper tract urothelial, looking at EV plus Pembro in a neoadjuvant setting. There's another study looking at, now more in our world, non-muscle invasive disease, intravesical installation of Enfortumab in bladder cancer. And there's evidence to suggest that non-muscle invasive bladder cancer is more luminal in nature than muscle invasive. And so theoretically, these tumors should be very sensitive to EV. But we don't know if it works if you just put it into the bladder. So that's something that's being investigated right now. And then there's also another neoadjuvant study looking at EV plus pembrolizumab in the neoadjuvant and adjuvant setting for muscle invasive and locally advanced. So, these drugs are coming down the pipeline very, very quickly. We'll be seeing them soon in clinic. And we're really trying to build as Vadim says, a Nectin-4 center of excellence here. So conclusions, part 2, ADCs are novel therapies here to stay in urothelial cancer. acquired resistance mechanisms are highly variable and likely patient and tumor specific. We got to think about both the antigen and the payload. And large-scale CRISPR screens may identify shared or conserved pathways of resistance and allow us to develop future drug targets or ways to modulate the sensitivity to existing therapies. And again, predictive biomarkers are greatly needed. And I think, when you think about these drugs in a third line setting, you're okay, throw the kitchen sink, like anything will work. When you start moving these things earlier and you start thinking about having to get these patients to surgery, that risk benefit balance does shift a little bit. And so, the EV plus pembro is very active. It works, it's going to replace chemotherapy in the first line setting, but there is a fair amount of toxicity associated with the treatment. And, rash is a big one. It's like 80% of patients now. All the immune related adverse effects, which, you guys have all seen, which are rare, but can have very long-term consequences for patients. So, I think, we need to see kind of, particularly, if a patient comes with muscle invasive cancer, they have now potentially different options for figuring out what kind of neoadjuvant therapy they can get. Is there a way to look at their initial TUR specimen to say, this patient will be very sensitive to one treatment or another? So those are things that we really need to continue to work on. And so one of the visions I have for work here, and one of my very close friends and collaborators now, Cornelia Ding, is involved in this, is to build these tissue microarrays and to do in a very focused way to look at, these matched pairs. So, if there's a patient who underwent surgery, has a tumor in the primary, a tumor in a lymph node, that's a very valuable way for us to study heterogeneity and drug sensitivity. So doing some of those match pairs. And in collaboration with our medical oncologists, which they've already been doing, on their own, is to get research biopsies on patients who progress so that we have very clean samples of what's happening pre and post treatment. This is valuable tissue for collaboration. It can be validated prospectively in clinical trial. And it allows us to do whatever testing we want on the platform of the day. So right now there's a lot of interest in pathology, AI on H&E slides, and whether or not just looking at the H&E is predictive of any particular outcome. A tissue microarray allows us to do things like that. And then, the wet lab component would be to continue to refine our models to test mechanisms of resistance. And again, the question is what sensitizes to EV? One thing I've been thinking about is how do we demonstrate a bystander effect? If anyone has any ideas, feel free to join the lab. And so I think, there's a lot of really exciting things that we can look at. And so just want to introduce you guys all now to my lab. Sasha just joined. She is an MD PhD. She was trained in China as well as the Netherlands. She's going to be working on STEAP1, which is another targetable surface protein actually in prostate cancer. Didn't have the chance to talk about that today, and doing some prostate cancer work. And she'll be applying for residency in the next few years. And, hopefully you guys will see her around. Jenna, who you guys know, is SUO fellow first year in her research year. She's helping develop the targeted CRISPR screen that we talked about earlier. Kevin, I put him in my lab because he technically sits in my lab, but a lot of the work he's done has really been independent. And he's going to be helping design the CRISPR screen as well. So he's moving into that world a little, as well as continuing his CAR-T work. And he's mentored by John for that work there. And then now I have two, incredible medical students who are also interested in working together. We're doing a mix of clinical projects. Ariel there is interested in working on this Artera AI classifier for BCG response in non-muscle invasive disease, and that's probably a whole nother talk. So yeah, we're excited. We're just starting, and I also want to highlight our collaborators up there as well. Felix Fang, John Chow, David Solid at Memorial Sloan Kettering, Cornelia in pathology, and of course, Seema Porton. I say collaborators, but they're more my mentors. So lastly, I want to just thank the team. I would not be here without you. Dr. Carroll, Dr. Fay, they are really the reason why I'm here, and and have such tireless purpose to everything they do. And I just felt, this is UCSF is where I need to be to continue some of this work. John, who taught me how to think like a scientist and still does. Dr. Breyer for his support of this surgeon scientist path and all his leadership with the department, and everybody here who's played a role in my development. I want to thank you all, and all of my collaborators at Memorial Sloan Kettering in the medical oncology department, urology department. My funding, so startup funding, hopefully we'll make some good use of that in the next few years, and then other sources of funding listed there as well. And we're a family here, so I also want to thank my family. Here's Parker, my 2-year-old, who's designing his own CRISPR screen of sorts. And my husband, you can't quite see him, but he's watching him back there. He's completely tired out and on the couch, and literal and figurative rock in my life. And then my family over there to the right with all our dogs and in Tahoe. So with that, we have a few minutes for questions, and thank you all for your attention.
Carissa, Will, really, what a wonderful talk. And I know if I can follow it, you did a really good job for the audience. So what goes into building these tissue microarrays? You said that's one of the goals.
It's actually, a lot of the onus is on pathology. So you just have to have a really good path colleague. So it's a matter of just identifying the patients and then having the pathologist go back to the original tissue and doing sort of punches of the blocks to then build this array. So you can tell them, by accession number, I'm interested in this patient. I want this lymph node, I want this primary tumor, and then you'll go and look at the block and kind of see where the areas of tumor are most enriched. And so it's really, we have TMAs in bladder and prostate as well, and I want to build one that's pre and post and this one is not.
I think this is such an exciting talk, from a molecular biology standpoint, but I was also really curious from an access to care and an access to these 2 drugs.
Yeah.
Are these used off-label, or is there funding from CMS for these, or how does the pathway look?
So they're like EV plus Pembro. So the combination treatment right now, I think, runs about $40,000.
Per year or per month or per what?
Per infusion. Yeah. So it's kind of a lot. I didn't really get into too much, but part of that kind of impetus for me doing biomarker work is also, as we expand the pool of people who can get it, there's always the access question, who's actually getting it, but also can you prioritize the patients who respond the most? Right now, everything's sort of given in clinical trial setting because it's not FDA approved yet in the first line. It is in the third line for monotherapy. So yes, it's
Great talk. I only understand about 1%, so I can only ask one question. First of all, I like your Valentine Fellowship because I got that.
Oh, you did?
I got that to come here.
Oh,
cool. So your antibody conjugate. So basically, you try to do 2 targets, right? You want to get it in, and you want to break down the nectin form.
yeah.
Now, you also mentioned the CRISPR-I. We have done some CRISPR-I in the lab, and it actually work out quite well. We inhibit myostatin, and the muscle get bigger, like a Jim Smith.
Oh, yeah.
So why don't you use the CRISPR-I?
Yeah,
inhibit
that will be.
nectin.
We'll do a CRISPR-I screen.
Mm-hmm.
Yeah, not that screen.
And if you do that, do you do a multiple CRISPR-I or just one?
So
2?
yeah, great question. I actually didn't mention. So what we'll do is we'll actually do it in different cell lines, kind of thinking about them as different patients almost, and seeing what are the similarities and differences when we do the same screen but on different cell lines.
And how are you going to get CRISPR-I to the body? Do you have any idea?
Oh, sorry, this is actually just a preclinical
I know,
work,
yeah.
so we won't use it in people yet.
Mhm.
There are things that we, an interesting thing is, rosy glycan. It actually is a PPAR GABA agonist and will increase Nectin-4 expression.
Which one?
Rosy glycan.
Uh-huh.
Yeah, so John's done some work on that in the lab, and he's able to make cells more sensitive just by giving existing drugs.
Great.
Yeah, I think, or the CRISPR stuff is just a screen. It's just a platform for us to look at, mediators of sensitivity and resistance, but it's not ready for use in people yet. Yeah.
I speak for everybody when I say we're so proud of you and so excited to have you back.
Thank you.
And I want to take a step back and ask you, when you were starting residency and when you were sitting in the shoes of many here, how did you set yourself up for your research year? How did you take advantage of mentorship in your research year to kind of launch this amazing vision for your career?
I think that's a good question. I think you just have to try, and I think you actually. So, like when you tell a story in hindsight, it seems like such a clean line, but actually, it's really not. I think I tried a lot. I had amazing mentors. You kind of chase the opportunity. I listed the grants I got, but I didn't list the grants I didn't get. So I think you just have to take advantage of all the opportunities here. Don't be afraid to try different things. I actually wasn't sure I was going to do oncology until towards the end of my 5th year. And, had the chance to be mentored on a lot of projects with a lot of different people. And just go with your gut. Don't be afraid to try something different. And it's really about the process that matters. It's not really kind of like what paper you're going to get out of this or where the paper is going to go. I think as a trainee, you're trying to think about what do I like, what's enjoyable, what can I spend my nights and weekends doing, how do I chase that, and then eventually you don't feel like you're making progress in the moment, but then eventually you kind of end up having like, oh, I guess this is my thing now. Yeah. Yeah, not advice. But also you come join the lab. We need people.
I have 2 questions. One, has anyone ever looked at like the timing of EV pembro in terms of like EV potentially? The first question is like, what do they do clinically currently in terms of like which one did they give first? And I'd be curious to see if there'd be any benefit of potentially like antigen presentation of doing EV first.
Yeah.
Maybe we can start with that. I have one other question.
Yeah, it is given in kind of like separate settings, I believe, and it does seem that So the real answer to your question is we don't really know, and part of it is like the drug work, the combination works. It works really well, so we'll probably continue to use it. And part of this research interest is to kind of answer some of those questions in preclinical studies. And so that's kind of like why that interest in that bystander effect is interesting. It does seem to be, so the other thing that's been observed is that MMAE itself seems to be more immunogenic than other payloads. There's a lot of interest in conjugating MMAE to other, antibodies like HER2, so on. So that may be a good one, like that may be the one to focus on. The actual mechanism of why and whether or not we give pembro later will make a difference or not, we don't know.
And then I guess just another question is, I know you're doing the CRISPR screen and cell lines first as like a proof of concept, identifying like potentially like innate mechanisms of drug resistance. I know like at MSK at least, like Poirier, I don't know if Poirier, I think is how you pronounce it, but his group is actually doing like functional CRISPR screening in vivo, in like xenograft models. I think that would be like something really cool. Like future direction.
Yeah, for sure. I think so. I think there's no model is perfect, but that may be, at least if you have a focused way of doing it, where you're not doing like a whole genome, but you've got a good list of candidate genes. Maybe you find that in cell lines. You can use that for that.
But amazing talk, yeah, I was really inspired.
Thank you. Thank you.